This last time we were doing SVM, then we can move to PCA. So basically, it is very important to have a very good SVD solver because everybody will do PCA. And when I say everybody, I mean everybody. If you take a random scientist out in the world, there is a 90% chance that they use PCA for something, okay? So you take people in machine learning, you take people in bio, you take people in DNA processing, you take bioinformatics, everybody uses PCA, okay? It's probably the most successful numerical algorithm that of the century, right? So something very, very simple, but it's something very it's something very, very widespread because the type of insights that they give, they spread across many different areas. Okay, so let's say some of the uses of PCA, right? So here they use PCA in order to do this, right? They represent the matrix in which describes the food consumption in the UK. You see on the row level, you have the type of foods. I don't know why alcoholic drinks is first. We know that alcohol has calories, but I will not call it a food. Anyway, also don't ask me what is carcass meat. Carcass meat? I have no idea. Anyway, so the strange British food on the row, and on the columns you have the different parts of the British Isles, England, Northern England, Scotland, and Wales. And you can see here that this is represented as a matrix, meaning that the entries are represented as a bar plot. So you can imagine this as a matrix, simply as a matrix, right? So if you had to do PCA here, you can ask yourself what are the type of information that you can extract by doing PCA here, right? So PCA will project this into a set of eigenfunctions, right? And you can ask yourself, what are the eigenfunctions? So the question is, how can you focus on just a few variables, right? So the matrix that we have before, it's a very large matrix and contains a lot of information. And as humans, we like to focus on a few components at the time, on two-dimensional components at the time. So what we do here is that we do SVD, right? From SVD, we extract the eigenvectors and eigenvalues, left and right eigenvalues that corresponds to the largest eigen. Left and right eigenvectors that correspond to the largest eigenvalue. And you will see that we will have a PCA component that contains this, okay? So the right eigenvector will contain this, will contain the amounts of food, right, that are consumed in Northern Ireland, Scotland, and Wales, and England. Okay, so here we will have the variables, right, and here we will have the regions. Okay. Also, right, we can do PCA on the other dimension, right, and we can say that the PCA1 is obtained as this linear combination of the foods, right? So here you see how the PC1 is comprised. So here we have the alcohol, the beverages, the meat, the cereal, the cheese, okay? So how do you think about this object, right? You think about this object as follows. There is some sort of prototypical, right? There is a prototypical individual. There is a prototypical British person that has this specific diet. Not much alcohol, not much fresh fruits. And not much fruit. A lot of potatoes, and a lot of soft drinks. And this is some sort of prototypical food consumer. And then this representation, right, is telling you how many of these prototypical users you will find in the different regions of England. Okay. So, for example, the largest negative entries correspond to the alcohol and the fresh fruit. The largest positive entries correspond to potatoes and soft drink. And so you can see that the information in the table is decomposed here in sort of populations, right? Subset of populations. And you match the population to a certain diet. and then the diet, and then the other part of the matrix tells you how many people from this population can be found in the different regions of England. Okay, so it takes a while to believe, right, that this is the correct way of that this is a meaningful way and the correct way to represent the data. But basically, this is why PCA it is so useful, right? Again, so the matrix is here, right? So you understand, you imagine that you decompose the matrix into the left eigenvectors, your diagonal matrix that contains the eigenvalues, and then the right eigenvectors. Now you have to imagine yourself that you have ordered the components so that the largest eigenvalues appears on the top left of the diagonal matrix, so that the left and right vectors they have been reordered accordingly. Now you have to yourself, what happens? Like, what is the meaning of the first PCA component, right? Because the first PCA component is associated with a left eigenvectors and a right eigenvector. And the left eigenvectors, we have this one here, that we have the foods. And on the right eigenvectors, we have the regions of England. Okay, so this eigenvalue corresponds to a specific diet, which is this one here, and to a specific frequency of this population occurring in the 4 regions of England. So if you cut, for example, the representation of this table to the 3 largest eigenvalues, 4 largest eigenvalues, then you understand that you have decomposed the information of the matrix as the sum of 4 outer matrices. And these 4 outer matrices, they will represent the food consumption of a certain population, right? There has this geographical distribution and this specific diet. Okay, so this is why so this is the beauty of PCA, and this is how you very simply turn an abstract problem that you see in linear algebra, talking about eigenvectors and eigenvalues, into some very powerful insight on your data representation. Okay. So just a couple of comments here, right? So the eigenvalue, you can incorporate it in either of the 2 components. You just have to decide how you're going to do that. So you can incorporate all on the left or on the right, or in both. That depends on the type of decomposition that you want to do. In this case, it appears that they have chosen to put it on the population, right? So this is the real population, is not the approximate population. And you notice again here the discussion that we were having in the challenge with NMF, because you see here that your food consumption, they have negative values. Okay, so of course it's not very meaningful to say that you eat minus 0.5 alcoholic drinks, right? So this is the interpretation that you have this when you do PCA, is that there is a prototypical consumer, right, which is your average consumer. And so the consumer in this population consumes minus 0.5 alcoholic drinks with respect to the average consumer. Okay, so it is still meaningful. But of course, if you had the decomposition which only contained positive values, then you will be happier, because this is, you wouldn't have to have this regression to the mean, let's say. Very good. So now let's say that we decide to a different representation. And what we do is as follows. We decide that the matrix is approximated only to the two PCA components, PCA component 1 and PCA component 2. And what we do, right, is that we want to represent the regions in these 2 PCA, with respect to these 2 PCA components. Okay, so this is a scatter plot. And this is telling you, right, that for the Scotland, right, there are this many of this prototypical food consumer of region 2, and this many from from consumer from group 1. Okay, so you see that they are pretty imbalanced, right? So this guy only appears in PCA 2. It does not. Sorry, the blue one, right? This population, right, that corresponds to PCA component 1 does not appear in PCA component 2, right? So in England, you only have the PCA component 1. You don't have PCA component 2. Yeah, so Northern Ireland is here. Yeah, so you understand that this is another way of representing the prevalence of one or two PCA components in the different regions. And so what you can do now is that you can compare the left eigenvector of these two, of PCA component 1 and PCA component 2, and you can see, for example, where they differ the most. So you see that this guy has a positive alcohol consumption, and this guy has a negative alcohol consumption with respect to the mean. So the alcoholic drinks is a way in which the consumption of alcoholic drinks is an aspect in which you can distinguish whether a certain person belongs to prototypical food consumer 1 or prototypical food consumer 2. And then another big difference is here. Of course, is on the fresh fruit and fried potatoes, because this is very stereotypically British, guys, the British people, they only potatoes. And I like they also put fresh potatoes, it's not any type of potatoes, they are fresh potatoes. So you see here that there is a big difference in fresh potatoes. And the fresh fruit, you see here the fresh fruit, also the fresh fruit consumption is very different. So this guy has a positive fresh potatoes and negative fruit, and this guy has a slightly negative fresh fruit and a very negative fresh potatoes. Okay, so along these three variables is where we can distinguish PCA component 1 and PCA component 2 the most extensive. Okay, then another representation that we have, it's like here, where we put the different regions, right? And we can see where the prevalence is of component 1 or component 2, right? So very easy peasy. So let's do another example here, okay? Just to show you another context in which PCA is useful. So we have breast cancer data, okay? So we have tumor cell features, and we want to create a model that predict whether a breast cancer is benign or malign, right? Whether if the breast cancer is going to bring health problems or not. Okay. So we have 30 different cell features. So you can imagine that they take some of the tissue, and then they cultivate the cells, and then they describe the cell features from the swab. Okay, and there are many variables here that are very highly correlated. So if you want to try to determine a diagnostic tool which is based on the features that you observe, you will need to reduce the variable, right? Because the predictor should be minimal in some sense, right? You will want to be able to assess the minimum number of cell features in order to decide whether the cancer is benign or malign. Because, of course, if you have a lot of features in your predictors, it means that you have to measure a lot of features. These features are probably acquired by visual inspection, right? So you can imagine that there is maybe a nurse, maybe a doctor, maybe some technician that is there counting the cell features from the cell swab. And then the more features you use for the detection, the longer this is going to take. If you have a population of a few 100 million women that they need to do breast cancer screening once a year, then this will require a lot of resources, right? So it makes sense that you try to remove some of the features from the feature space so that you can have a very good prediction without stressing out your system. So this is your all set of features here. You see that the right and miss, they are correlated. So the first thing that you can do, you can imagine that what you do is that you just take a subset of the variable that they have significant correlation to the final decision. Right, so you see here that you go from 30 features to 10 features, and when you go down to 10 features, then you have dropped some of the features that you have manually predicted. And you see here that before we had both mean and standard variable. So you had 2 features per area. Now, if you remove the standard deviation and the worst case, right? Then in this case, if you do the count, you end up with only 10 features. And you can see here that you have simplified your test a lot, because now you went from 30 features to only 10 features, but you are losing some information with regard to what you dropped. So this is not ideal. So what you do is that you can actually be happier if you design a new data set. A new set of data, which are linear combination of the original features in whatever coefficient you have. Okay, so you start with these 30 features, and then you say, well, I want to have only 3 features, and I want these 3 features to be a linear combination of my previous features. And I want to have this feature designed in a way that they can still be very effective for prediction. Okay? So the idea now is to use PCA, and we will use PCA to obtain this combination of variable. And by looking at the eigenvalues of PCA, we can decide to reduce all those components that have very small variance, right? That they don't contribute to explaining the observation very significantly. And we can just keep the highest PCA components. And these high PCA components will be given to us as the linear combination of past components. So we will be able to sort of retain an idea of interpretability of how these new variables were obtained. But of course, since we start with 30 predictors, if I give you a linear combination of 30 features, the interpretability that you have over this linear combination is limited, right? Especially if we start also having positive and negative signs, right? So very, very tricky. So, but if we do PCA, right, then that's what we obtain. So let's say that we have 6 patients, and we just look at the 2 features at the beginning, right? So we just look at the radius of the cells as a mean and the smoothness of the mean. And here we do a scatter plot, but we color code it whether it was malign or benign. So this is the value of the scatter plot. So you see very well that here there is a very clear separation between benign and malign, right? So you see that with these variables, you have a good discriminative power of what is malign and what is benign. So what you will do here is that you will determine what is the center of the data set in terms of mean and value of each of the features. So the smoothness is roughly around here, and the radius is roughly around here. You see that roughly they are balanced. And you will use, then you will shift the data set to the center of this object. So now we have we have the mean, right? We have moved this scatter plot to the mean. Now we find the straight line that passes through the fitting of the data set. So we do the fitting with respect to the full data set, both malign and benign. And then what we do is that we compute the distance of each of the points of the data set to this line. And this is the projection of the PCA component, right? So we project this point to this point, these 2 points to these 2 points. And then this is the PCA component 1, and the PCA component 2 will be the distance from the center. Okay? So then this is this reframing of the PCA components in terms of the projection of the two components. Okay, so they are orthogonal. So here we're looking at only 2 PCA components. We know the PCA components are orthogonal, right? So if this is the first PCA components, we are only in dimension 2. Then the second PCA components might be orthogonal to this one here. Since on a plane we only have 2 orthogonal lines. This is the PCA component 1, and this is the PCA component 2. Okay, good. So here you see what happens if you change the way in which you have done the curve fitting, right? So here, so here, what have we done here? Here there is the outlier, right? So if you move the outlier is the same. Okay, so if you change it's telling you what happens if you change the fitting curve, right? What is the effect of this? Second. Then, okay, so this is the data set that you have. What you do is that you look at the covariance matrix and you look at the covariance matrix of this object, and this will give you an indication of how sure you can be of your prediction. So the covariance matrix of the matrix will be 1 over n minus 1 A transpose A. So this is just following the definition of covariance matrix in data set. Remember that we have 6 individuals, so the computation of the covariance is done by this matrix multiplication. Then what we do is that we diagonalize the covariance matrix. So this X transpose A equal to X dX transpose, where the X are the eigenvector. This is a diagonal matrix with positive elements, right? So that we know that this is going to be an SVD. Then if we do SVD over this, we will obtain U sigma V transpose. And the maximum variance, so the largest variability of the elements, will be corresponding to the large singular value. Okay, so basically the first PCA components is the component that which has the largest that explains the largest part of the variability in the data. In the sense that if this is the data, and we look for the dimension in which the variance of the data is maximized, the variance is maximized over the direction of the first PCA components. Okay, so the first PCA components, if you have to remember it with one sentence, is the direction that sees the maximum variance. And then the other component is the orthogonal component, and the PCA component one is this guy here. The value of the points in the PCA component one are the projection of these points in the scatter plot of this component, and the PCA component 2 will be the orthogonal component to PCA 1, because by construction, and then the projection of the points on the PCA component will be the other corresponding eigenvector to the PCA component. Very good. Now we have a transform data set in which we can represent the components. We can represent the data set entries in terms of the projection of the two PCA components. The word is all right with us. So now we have malignant and benignant over these PCA components 1 and PCA components 2. And now we can again have a simple test that discriminates between the two variables of this reduced space. Okay, so even if we started with 100,000 features, then these two features will boil down to be PCA component 1 and PCA component 2. Very good. Let's do one more example, which is the flower classification. So you're given a flower and you have different measurements of the flower. Characteristics. I don't know if there is a better name for this. And then you have different labels. Okay, so you can have these setosa things, this versicolor, this virginica. Don't ask me what type of flowers are. I'm not a flower expert yet. Then what do we do is that we do this plot, right, where we plot the following thing. So on the x-axis, we plot the histogram of the feature. So we look at the petal width, the petal length, at the sepal width and the sepal length, and we do the histogram of these values under the different species that we have. So we have 3 species, and then on each of the ij position of this subgraph, we put the scatter plot of feature I versus feature j, right? So in this case here, we have the petal length versus the petal width. And you see that there is some very nice clustering where we have that the setosa has a small petal width and small petal length versus the virginica that has a large petal length and a large petal width. Okay, okay, good. So now the data set still has a lot of features. We want to have fewer features and still be able to distinguish the 3 different flowers. So what do? It's our usual trick. We do PCA decomposition. We obtain a new set of variables, and we use this set of variables in order to decide whether we are in the presence of this setosa, this virginica, or this, what's the third one? This versicolor. Okay. Again, the trick is the same. We take the data set, we demean the data set, right? So we project the data set into the mean. Then we compute the SVD as U sigma V transpose. And then we look at the principal components using SV decomposition. And we find the principal direction, which are the columns of V. Then we cut down this principal direction to a certain number that we like, and we call this the A-star dataset. Okay. So now the variances to the new dataset correspond to the singular value squared of the original datasets. So you can see it here. A star transpose A is VT ATA times AV. We decompose this way, we decompose this one, and this is equal to sigma transpose times sigma. So the variances of the new components is the product of the diagonal matrix in the SVD decomposition. And if we want to, so the purpose of this A star is to have a smaller dimension than the original one, because we want to have fewer dimensions, fewer variables to use for our decision. So we choose a reduction to dimension k. So then we will have that this A star, right, which is equal to m times k. The A is the original A, which is m times n. The V is this projection matrix over the first k PCA components. So this is n times k. Okay, and then if you combine the dimensions, and everything goes well. Okay, so this is the data set. You can load it up and you can have fun with it yourselves. Then the last concept is the explained variance, right? Meaning that if you cut the representation of your matrix after k components, how much are you leaving on the table? How much error are you committing between your A star and your A? Okay, between your approximated matrix and the true matrix. And usually here we use the explained variance representation, where we look at this. We look at the variance which of the ith component divided by the variance of all the other components, because by the law of nature, you know, the PCA component is the largest one and all the other components are small. So if you include PCA component 1 only, then you of the total variance that you have, you explain the variance of the first component divided by all of the other variances, right? So in this case, this was 75%, almost 75% of the total variance. So it means that if we apply, if we only preserve the first PCA components, then we have 75% of the variance. And you see that PCA components 2, let's say it has 21% of the variance. So if we take PCA component 1 and PCA component 2, then we explain more than 90% of the variance. And so we know that if we take k equal to 2, then we're explaining 92% of all of the variance. Okay, so this formula it's a percentage formula. Okay, so now we know that we only need one PCA component because we already explained most of the variance. So this is our PCA component 1. And what we do is that we plot the projection of the features into the PCA into the eigenvector corresponding to PCA component 1. And we see that a very clear clustering emergence. Sorry, guys. So the setosa is here separated, and the versicolor and the virginica, they are partially separated, right? We can partially separate the two of them. Very good. And now we plot both PCA component 1 and PCA component 2. And now you see that there are more clustering emergence, right? So we have higher dimensions, so we can see clusters better. And again, we see that this setosa is very easy to classify out. But this versicolor and this virginica, they have a part in which they still superpose. Good, then we can go and look at PCA component 1 and PCA component 2 and see which features do they correspond to. So this is the petal length, and this is the sepal width. So this is the representation of the flower in terms of the features. And so let's go back to this data set with a lot of features. Let's say now we are back to this cancer. So now in that case, the explained variance grows much slower because we have more variables, so there are more opportunities for the explained variance to be spread out over multiple variables. So this is the cumulative variance that you can, the cumulative explained variance of your data. Here again is the clustering according to PCA1 and PCA2 for malignant versus benignant. Here again this plot that gives you malignant versus benignant in the different PCA component one. And now, since you're representing things in the PCA components, you can ask yourself if the PCA components reveal any specific cluster. So you see that PCA0 and PCA1 can be used to discriminate well. PCA1 and PCA2 not so well. And also it seems that PCA2 and PCA3, they're too close. But for example, PCA2 and PCA1, they seem to be good linear combination of features to distinguish benignant versus malignant. And so we can see that the P0 determines this much of the features of the data, and this is it. Okay, so basically the message here is that PCA is very, very useful, and you guys will have to use it for sure at some point in your life soon, beyond my challenge. Okay, so let's have a very quick break, I think. Let me just, maybe we discuss the, just a second, guys, let me think. Okay, let's have a quick break and then we talk about PCA a little bit more, and then I'll let you guys go, okay? Okay, ciao guys, 5 minutes, ciao guys, 5 minutes, I'll see you in 3 minutes. At 25, I'll see you back here, okay? 3 minutes, go walk around, take a break. Okay, guys, let's discuss PCA a little bit more from a different slide set, then I'll let you guys go. So again, as we said, the way to imagine PCA component is again in the 2D dimension, because we are built for 2D dimensions. So you imagine that you have a scatter plot of points, and on this scatter plot of points, each point is represented by 2 coordinates, right? So you want to re-parameterize this scatter point by changing the reference axis. Okay, so you want to choose the first axis to be the direction in which you observe the largest variation of the data, and the second axis is the axis which is perpendicular to the first axis. Okay, so again, the idea of the first PCA components is the direction over which you observe the large variance of the data. Okay, very good. So the idea is that you start with a data set, and after you apply PCA, you obtain a set of components. The number of components that you obtain is equal to the original cardinality of the features of the data set, which is Z1 to ZP. Okay, so each ZI is a linear combination of the original predictor, and usually you can normalize the components to have a weight 1, norm 1. So this is depends a little bit on the application that you have. As we said, the orthogonality is an important feature. The fact that they have norm 1 can be a little bit skewed or adjusted according to the application. Then all the Z1 to ZP are orthogonal. Okay, so they are pairwise orthogonal, and they are ordered from the largest variance to the smallest variance, explained variance. And when we want to reduce the dimensionality of the data, we pick a certain number m, which is the number of principal components that we want, and then we re-express our data as the linear combination of these m components that we will have, and this is a reduced rank representation of the data. Okay, so the intuition is this one here. You want to capture the most amount of variation. And why do you want to do that? Because data has to vary along interesting directions. So there is the idea that the direction in which the data changes the most is where the most interesting effects happen, right? So you can imagine that if the noise caused the largest variation, then the noise will dominate data. So if we imagine that that this is the case, then the largest variation would be in the presence of information. And why do we want to enforce orthogonality? Because in a sense, we want these explanatory variables to be orthogonal to each other, right? And so if they were correlated with each other, then it would be uninteresting for us, because we will have multiple components that try to explain the same underlying variability in the data. Okay, so the orthogonality is very strong, and you can imagine that you transform the original points into the original cloud points in the scatter plot in the following representation. There is the PCA component here, and then there is the projection of the points in the scatter plot into the PCA components, so that each point along this line is obtained as a linear combination of the points of the two PCA features, and the linear combination is according to the equation of the line over which you have projected them. And then the second PCA component is the distance, right, from this point to this line. And remember that the distance between the point and the line measures the orthogonal direction. So everything goes as explained. So if you decide that you want to represent the original data into these p dimensions, what you're saying is that the value of the components is this matrix of weights omega p times omega p multiplied by the original data x. Okay. So then your matrix x is the original data matrix, which is n times p. And the omega is a P times P matrix in which over the columns you are the eigenvector of the square matrix X transpose X. Okay. Remember that you can decompose according to the left eigenvectors or the right eigenvectors. So you have to also decide a little bit how, which dimension you want to decompose, right? So we saw the example in the diet that sometimes we decompose them as prototypical diet and sometimes in terms of presence in the different region of England. Okay? So the important step that you never have to forget is that when you have to take when you take your original data, you want to apply PCA, you always have to standardize the prediction, standardize the predictors to have zero mean and unitary variance. So there is this scatter plot of points, and you need to put this object in the middle of your representation, right? And you need to normalize the variance to 1. If you don't do it, right, then the PCA components will contain information regarding the shift. Okay, but this is not interesting because these are constant components that are not relevant to the variability of the data, right? Then you calculate the eigenvector of the matrix X transpose X, and then you obtain the eigenvalues of this object. These eigenvalues will be in this matrix omega, and then you multiply your data with your matrix omega, and in this case you will write multipliers. Okay, if you want to take the left eigenvectors, then you consider X times X transpose, and then you will do omega times X to obtain your component. Okay, so in Python you do it as a decomposition.pca, because as you can imagine, PCA is very sensitive to the presence of very small eigenvalues, to very large condition numbers. So there are many different numerical methods in order to do the PCA decomposition. And if you just let Python do his job, then it will contain a lot of routines that evaluate which approach is the best approach for PCA decomposition. So in this case, we're taking the data where you have a certain person, age, the cardiac speed, the cholesterol, the max heart rate, and what you're doing is you're predicting whether they have subject to a heart attack. And then when you do the composition, Python will give you what is the first component, the second component, and then it will give you the variance which is explained, the variance of the data which is explained. Okay? So remember that this explained variance is unsupervised type of number, right? So both in the example before, in the example now, we are doing some classification, but the variance that you observe here is just variance in the data. That is not with respect. Sorry, it's not variance with respect to the classification task. So here we have again the heart rate yes, and the heart rate the heart attack yes, the heart attack no. We represent the scatter plot now in the two PCA component 1 and PCA component 2, and you notice that the two PCA components don't explain much who has a heart attack and who doesn't have a heart attack, but of course we can try to draw some type of conclusions, the type of the size or for the classification task, taking into account pulse rate alarm and missed probabilities and so on and so forth. Okay. So the example that we saw so far were for classification. So what we wanted to do was to separate the data so that we can, we want to represent the data in a small dimension so that we can have a separator, which is a simple separator between class 1 and class 2. But what we can also do is that we can do regression. Okay, so we can try to use it for performing regression. So in this case, we might want to reduce the dimensionality in order to simplify the regression. We can choose a small value of m in order to have a more robust predictor. And and then we will have some coefficients for this prediction, which we can validate using cross-validation. Let's see some notes about this. So PCA is an unsupervised algorithm. Okay, it means that it's a way of representing the data in a lower dimension in order for us as humans with two eyes living in 3D space to observe some pattern which is present in the data. And this is regardless of what is the task for which the data is being used for. Okay, so the fact that you try to order the data from the one for the dimension that has the most variability to the one that has the least variability, it doesn't tell you that the variability takes place in the dimension which is relevant for your downstream task. Okay, it's just a hope, right, that if you look at the representation, then this representation is going to preserve most of the information which is present in the data. But of course, this is not always the case. That this is what happens for your downstream task. So a classical example of this is genetic disease, or let's say genetic analysis. Okay, so if you take the DNA, right, the DNA is a very large dimension, and so you do not want to work with DNA sequences, right? So if you look at DNA sequences, there's not much for you to understand because they're very long and they are on alphabet 4, right? So very hard for us to make sense of it. So what you do is that you take, let's say, you take the DNA of a large set of people, let's say a few 100,000 people. You represent it as PCA components, and in these PCA components, you find some useful representation that says, for example, the presence of certain genes or the presence of the expression level of some genes. So you can reconduce the PCA component to some understood genetic mechanism, and then if you look at the PCA components from the one that are most explanatory to the least explanatory, and you're looking for some specific genetic disease or specific population, or let's say a level of closeness between people, then you will have to discard the top PCA components because the largest genetic variabilities have to do with something like, for example, color of the skin, right? Or male and females, of course. Some very large genetic traits, right? So the largest variability of the DNA is in genetic traits that are uninteresting for you, right? So if you disregard these largest components, then you have to with a smaller number of components that have a smaller explanation of the variability of the DNA sequence. But this is where the information that you're looking for is, right? Because let's say gender, height, I don't know, feet size, weight, BMI, whatever, they are not interesting, right? So they explain the largest variability in the genetic information, but it's not interesting for us. Good. Then what are some of these disadvantages of PCA? Is that the interpretation is lost to us because these components, they contain a linear combination of maybe thousands of features. So how much interpretability is left is debatable, okay? So maybe if you want to have something particularly interpretable, then you have to limit the number of non-zero components in your PCA, components. And this will, of course, introduce larger complexity. You will have to regularize things. You will have to add, for example, an L1 norm into your PCA decomposition, and this will complicate, let's say, the efficacy of the use of the technique. So it's very good for reducing dimensionality. It's very great for visualizing the predictivity of the features. And since we impose the components to be orthogonal, then it's great for reducing collinearity, okay? So let's say you have a lot of data, and what is easy for us is to observe pairwise correlation across features. But it's very hard for us to see if there are, for example, two features that fully predict a third feature. Okay, from a point of view of linear algebra, we will see this in terms of the rank of the matrix or of a specific submatrix, but of course, it's very hard for us to see this. But if you represent the data as along its PCA components, then each component is orthogonal, so collinearities are easier to eliminate in the reduced representation. Right, sorry. Yeah, so this is regarding interpretability, right? So what you can do is that you can look at the PCA components, how they're obtained as a linear combination, and just pick the largest contributors to the components and say, oh, this component is dominated by this specific feature. yeah, and then what you can do, for example, is that you can see what happens to the weights of a. So this is the experiment that we're doing, right? So we're doing PCA components, and we are seeing at how much each component is each of the original components is present in the different PCA components, right? So you see here that as we add more and more components, the composition of the different PCA components that changes proportionally. So there is not much continuity, right? So if you do PCA with different number of components, then the relevance of the different components might not be preserved from one K to the next K. Yeah, so sometimes you want to add some regularization in order to improve the smoothness. And yeah, that's about it. Okay, guys, so I think this is enough for today regarding PCA components. Next time we'll see the presentation, so I think it'll be very nice. I think the guys, the work that you did so far was very nice. I cannot wait to see the presentation next time. So we said that we have 4 weeks, so I'll try to give you the final challenge next week. Then we do another challenge, and then I'll just we'll just go through some material very lightly while you guys work on your final challenge, right? So you will have 2 presentations that you have done as a unique group, choosing your own group, which is just one group, and then you will have the final challenge. If you have any ideas for the final challenge, let me know. I'm always happy to listen to your ideas for the final challenge. Otherwise, I will give you something that my brain can dream up regarding this. And yeah, so if you want to do some research during the summer, let me know. We have a few things that we're cooking up in order for you to work more on machine learning and deep learning, right? So the rate at which you acquire information regarding these topics in class, it's not as fast as if you learn by yourself. So we'll try to facilitate that. And otherwise, yeah, let me know if you have any questions, and I'll see you next time. We stop the recording so you can speak freely. We say hi to the people on YouTube. Ciao, guys on YouTube.